Imagine you've been handed the complete architectural blueprint for a human being. It's all there, but it's written in a language with 3 billion letters. For so long, we've had the book, but we couldn't actually read the instructions. Well, today, we're finally decoding that blueprint.
So here's the real challenge, right? It's one thing to have all 3 billion letters of our genome sequenced. It's another thing entirely to find the parts that actually do something. It's kind of like searching a massive library full of books with the same 3 billion word story. And you're looking for these tiny critical sticky notes in the margins that tell you which sentences to read and when to read them. So how on earth do scientists find those crucial notes in all that text?
All right, so the first part of our mission is to find the genome's control panels, all the tiny little on-off switches that tell our genes when to fire up and when to shut down. And these things are absolutely essential for, well, pretty much everything. These switches have a name: transcription factor binding sites, or TFBS for short. The easiest way to think of them is like little docking stations. When a specific protein, called a transcription factor, comes along and docks on one of these sites, it flips a switch, telling the nearby gene to get to work. It's this precise network of switches that really orchestrates the entire symphony of life.
Yeah, but finding these sites? That's the hard part. It's a huge task. They are incredibly short, sometimes just a handful of letters long, and they're hidden inside this massive genome. So to find them, researchers can't just read the whole thing manually. They need really sophisticated computational scanners that are designed to spot these tiny, meaningful patterns in a vast sea of genomic noise.
So you've basically got two ways to tackle this. On one hand, you have the simpler scanners, like PWM models. They're pretty fast, easy to understand, but they kind of have blinders on. They can miss the bigger picture. Then, on the other hand, you have the AI powerhouses, like deep learning models. These smart scanners can spot incredibly complex patterns that the simple ones miss. But, and it's a big but, they need huge amounts of data and a ton of computational horsepower to run. It's that classic trade-off, right? Simplicity versus power. Okay, so we've got the switches, but the blueprint is also filled with these, well, abandoned structures, ghosts of old genes, that actually tell a really fascinating story about our evolution.
We call these things pseudogenes. At their core, they're just broken copies of real, working genes. And for decades, scientists just dismissed them as junk DNA, evolutionary dead ends, molecular fossils that didn't do anything anymore. But as we're about to see, that assumption turned out to be completely wrong.
So how does a gene even become a ghost? Well, one of the most common ways is through this funky process called retrotransposition. First, a gene gets copied into a messenger RNA molecule, an mRNA. Then, in this weird little cellular quirk, that mRNA gets reverse transcribed back into DNA and just randomly shoved somewhere else in the genome. It's like a photocopied page from a manual getting pasted into the wrong chapter of the book.
And this process gives us two main flavors of pseudogenes. You have the processed ones we just talked about, the ones that come from that mRNA copy-paste job. They lose key parts, like their on switch, which is called a promoter. Then you have duplicated pseudogenes. These are the result of a direct DNA copy-paste error. They usually keep their original structure, but they just get broken by mutations over time. But here's where it gets really cool. This is the plot twist. Some of these ghosts, they aren't dead at all. Scientists started discovering that some pseudogenes can actually be turned into RNA molecules that act like decoys or sponges, messing with the function of their original parent genes. So what we all thought was junk for so long is now seen as this hidden secret layer of genetic regulation. It's pretty amazing. Okay, so we can find the switches and we're starting to figure out these ghosts. But how do you put all of this information and everything else we're learning together into one single master map that every researcher on the planet can use?
Well, that's where something called the NCBI Eukaryotic Genome Annotation Pipeline comes in. Just think of it as the Google Maps project for the genome. Seriously, it's this massive automated system that takes a raw genome sequence and just systematically goes through and labels everything it can find. Genes, switches, pseudogenes, you name it. It's what creates the definitive reference map.
Now, the whole process is incredibly complex, but you can boil it down to 4 main stages. First, it masks out all the repetitive noise. Next, it aligns all the real-world experimental data we have and maps it onto the genome. Then, an AI program called Gnomon makes its best guess, predicting where all the genes are. And finally, and this part is absolutely critical, it curates everything, always prioritizing the stuff that's been verified by human experts over any automated predictions.
And this difference between human and machine is super important if you want to understand the map's reliability. The known RefSeq annotations, those are the high-confidence landmarks. They're backed by direct experimental evidence, and they get top priority. The model RefSeq annotations, on the other hand, are the AI-generated routes. You can spot them because they have an X in their name. They are incredibly useful, especially for parts of the genome we haven't studied much, but everyone knows they're predictions, not stone-cold certainties.
So, we have the tools to find switches, to study the ghosts in the machine, and to build this incredible master map. But let's take a step back. Why does all of this incredibly detailed work, decoding this blueprint, actually matter?
I mean, having a really well-annotated blueprint, it's a total game changer. It's what lets us pinpoint the exact mutations that cause diseases. It helps us understand the complex programming that makes a heart cell totally different from a brain cell. We can trace our own evolutionary history through these fossil genes. We're even uncovering brand new functions in DNA we used to call junk. Put simply, this map is the foundation for almost all modern biology.
And this really brings us to our final thought. Every single day, our map of the human genome gets more detailed, more accurate. The switches are being labeled, the ghosts are giving up their secrets, and as this ultimate blueprint of who we are comes into sharper and sharper focus, it leaves us with this incredible, tantalizing question. What profound mysteries about our health, our evolution, and our very identity will we finally be able to solve?